Hey, welcome back. This is Joel Duff, and today just a little behind the scenes, like other things that I do, like on my real job. My real job is not talking on YouTube, that's for sure. But what I really do is, I teach genetics and genetics lab and a variety of other classes at the University of Akron. And one of the classes I'm teaching this semester is, or overseeing this semester, is the genetics lab. This is a genetics course that is for biology majors, and almost all of the students in my lab are probably pre-med or think that they're pre-med students. And so I have 72 students in 6 labs, and I have 2 assistants that help me out in those labs. And I've prepared, we're doing a project for the whole semester that is a molecular biology experience. So rather than doing fruit flies and transmission genetics, like heredity stuff, we're doing more molecular genetics. And so it's a DNA project where they extract DNA, they use PCR to amplify a gene, and they take that gene and they cut it into pieces with restriction enzymes to create a restriction site profile, which is sort of like a little fingerprint in order to interpret what genotype they have and to figure out phenotype from that. And so they've been working through that through various steps in the semester. But on the side, they've also been learning about another human gene complex, which is what you see here, the human leukocyte antigen gene complex. And this is a really complex set of genes that function together to make a portion of your immune system. And so I've been having them learn about the genes themselves, the similarities of the genes, different variants of the genes, how important different genetic mutations are in those genes, something about the heredity of the whole system. And that kind of goes with the genetics class. And then I'm topping it off with this human leukocyte antigen bioinformatics assignment, which I've been working on for the last couple of weeks, and I'm ready to unveil it to the students in the next day or two. But actually, tomorrow morning. And so I need to make this. I need to make a video for them about sort of stepping them through the whole process. So I'll explain it in class, like briefly, and I have this whole set of instructions ready for them to go. That kind of steps them through the websites they're going to go to and what information they're supposed to look at. Then they have a worksheet to fill out, which is actually this multi-page thing that includes looking at the sequences and identifying their particular variant that they're supposed to find, and then developing primers for it. And well, I'm going to show you that in just a moment. So this is where they fill in all their answers. They download that file and fill it out and then upload it to where we can grade it for them. All right, so that's the introduction. Now what I'm going to do is I'm going to go into, in just a moment, the next slide is going to be my intro to the class, right? I'm talking to the class, I'm recording this for them. If they can't figure out what they're doing, right, I would like them to watch the video, because I think I'm going to explain more than they're going to learn by actually doing the exercise. But some won't be able to figure out everything they need to do just by reading the directions. But you know, how many students actually read directions these days? Very few. And so I know they're going to get lost. They're going to have questions. And rather than having to constantly answer the same question, I'm making this video so that I can say when they email me, did you watch the video? Because in the video, I explained like how to do that. And of course, if they ask me a question and I know I didn't answer it in the video, I will very kindly help them out. Or have one of my teaching assistants do so. All right, so human leukocyte antigen bioinformatics project. Got that coming up next. All right, we've been dealing with the human leukocyte antigen gene complex for many weeks. We've had some discussion forums. We've learned a whole bunch about that gene, and now we're going to put a little bit of what we learned in the lab to practice. All right. You've been using PCR primers and you've been using restriction enzymes in a designed lab. But what I'm going to have you do now is I want you to design a set of tools in order to do a genetic test yourself, right? So what you've been doing this semester is essentially a form of genetic testing. You want to know, do I have this variant or this variant of the ABO blood type? And but you've been guided through that process, right? Using somebody else's primers that they developed that they know would work, so that that you would get positive results, hopefully, right? As we're seeing this week. And But now what I want you to do is we're going to take another gene, the gene that we've been discussing mostly, which is the HLA-B gene. And I'm going to assign you a variant. So one of the first things you're going to do is you're going to pick a variant. So on the handout that I'm going to provide with the directions, it tells you, each student is supposed to select, what is on? Here we go. What variant have you been assigned or picked? So in class, I asked you to pick one of the variants, and here you'll circle that variant on the worksheet and say, like, here's the one you've been assigned, right, to find out something about. And you're going to take that variant, and in a nutshell, here's what you're going to be doing. You're going to find, first thing you need to do is find, like, where is this in the human genome, right? You know about the HLA complex, and I'm going to give a little intro again to remind you of the most pertinent information about the HLA-B gene in just a moment. But I think all of you recognize what that gene is and what it's probably important for. But now I'm going to have you find it in the human genome. I'm going to take you to the human genome browser, and I'm going to show you that in just a minute. And you're going to actually zoom in on that particular gene in a particular location, and you're going to hunt for this particular variant that I've assigned you to find. And once you find that variant, you'll discover things about it. I'm going to ask you a bunch of things about, like, what are the characteristics of this variant? What kind of mutation is this? And then I'm going to have you develop PCR primers to amplify, as if you're like, okay, well, what if I wanted to find that variant in me or you or anyone else? I want to make a genetic test for, do you have this particular version at this particular site, this particular change in your genome, or do you have this other version? How would you test for that? Well, one way to do that is, as you've learned, we could use PCR primers to go into the human genome, find that particular location, and amplify it out. So we have lots and lots of copies of it. Then we could take those copies and the easiest thing to do is, as we're discussing in class this week, is you could just sequence it, right? Just send it off to a company, have them sequence it and say, like, oh, here's that particular location that you're going to identify in the human genome. You say, you have an A there, or you have a T there, or you have a C there, or maybe the base pair is missing, or maybe you have an extra base pair there. That's one thing I want you to find out is what is the nature of these variants. So you're going to find out more about that variant. So you could just do sequencing, but I'm not going to make that assignment that simple, right? Oh, just send it off, get the sequence, and there's the answer. No, what I'm going to have you do is I'm going to have you develop PCR primers to go in and pull that thing out. And rather than just sequence it, I'm then going to direct you to how to find restriction enzymes that you will have to decide what enzyme actually cuts at that particular location. So it either cuts if you have the variant, or doesn't cut if you have a different version of that at that particular location. And from that, then, you'll be able to predict, just like we did in class, what your PCR fragment will get cut into, what size fragments. So when we run that on a gel, hypothetically, if you run your sample out on a gel and you had that variant, I want you to be able to predict what fragments you would see on that gel, what little bands would you see on the gel. And so that's part of this project too. And then once we find that, I also want you to explore the sequence in this particular region a little bit more. So I'm going to have you take some of that sequence out, and I'm going to have you take it to the National Center for Biotechnology Information, and you're going to blast it against all the other sequences, excluding human beings, because otherwise it'll be a really boring search, and find out how similar that sequence is to other organisms. All right, so that's the overall gist of the assignment. So now let's get into some details. So now I want to start by just really briefly, like, covering like what this gene is. All right, so we've been talking about the HLA complex. I think all of you should know it's on chromosome number 6. In fact, you should know that it's on the short end of chromosome number 6. And the HLA gene complex is a group of genes, a whole bunch of genes that have been divided into a number of different classes. See here, you have class 1, class 2, class 3. And you know that class 1 genes of the HLA complex are the ones that we've been thinking about, all right? A, B, C, and E, and so forth. And you know that most of those genes, that actually maybe you didn't realize this because maybe you didn't look closely at the structure of each gene. In some of the other assignments, many of you have discovered like the number of exons and introns and the size of the genes and like what their behavior is in the whole what they do as a product and so forth. But maybe you haven't actually thought about comparing those genes and how similar they are to each other. And I'll show you that on the next slide. But here we have HLA-A, the A gene, which consists of 8 different segments, 8 different exons, expressed sequences with their introns in between. And if you want to break that down further and get even, what we're doing is we'll just keep zooming in, because that's what you're going to do in the first part of your assignment. You're going to start with the whole gene. You find it in the genome. Then you're going to zoom into a small subsection of the gene. Then you have to zoom in even more till you get to the A, T, C's, and G's, right? The actual code itself. And here in this example, we have like, well, here's the overall structure of the gene, how the different exons are laid out, and how much space there is between them. And then if you zoom in on some section of that, you get to the actual code, and you see that there could be differences in that code, right? You know, you might have we all have this gene, but you might have 10 different locations where your sequence is different than mine. And if any of those are significant differences in terms of like making a different amino acid at that position, then we might identify that as being a different allele. We'd recognize it as being different enough that you have a different form of the HLA-B gene than I have. And of course, you got one from your mom, one from your dad, so you could have 2 different forms in you as well. And those 2 different forms could be different than my 2 different forms, because there are a lot of different alleles of the HLA-B. And so, but I want to, maybe something that hasn't come through in the class or the lab is that alleles aren't always just one single base pair change or one particular insertion or deletion that then constitutes the entire allele. Alleles are often haplotypes, and a haplotype would be a series of changes that are common, like, I have this base pair at position number 57, and I have this particular base pair at position number 103, and something else at a position 175, and I have this unique set of variants that a bunch of other people also have, probably because we share some ancestry, and that particular allele, that particular form of the gene has been passed down, right? And there's X number of those copies of the gene in the human population, so that's a particular allele in the human population. So that allele is multiple common mutations all linked together on one haplotype, one gene, all right, or one copy of the gene. And so how many different haplotypes are there, or alleles are there? There's some 703 down. I'm looking all the way down the bottom of this page here. 703 HLA-B recognized alleles in the human population. So that's a lot of different variants. So there's a good chance that my alleles, my parents gave me different alleles for HLA-B, and my alleles are different than both of your alleles. That's how different we can be in this particular gene. And of course, this gene, is part of the human leukocyte antigen complex, which is a part of your identification system and how you identify foreign invaders. And so we need lots of variation in this particular site in order to give us different opportunities to identify foreign products out there. Okay, so hey, I said I would do a really quick introduction. It's not feeling very quick. So let me I'll both speed up and shorten what I was going to say. I did want to show you the similarities between these different genes. So HLA-A is on the top, and then HLA-B, the one we're interested in, is second here. And HLA-C, and you see they all consist of like about the same number of exons. And if you were to line up their sequences are quite similar to one another. And they have the same number of introns, spacers between them. And sometimes the introns are even similar sizes. So this harkens back to something we talked about in class, which is gene duplications. It appears that these genes are ones that have tandemly copied themselves because they're right next to each other. So a gene has copied itself, and initially it would have been identical, but then mutations happen, which then make one gene different than another. And so we accumulate varieties of these genes, right, which we now call different genes, or a complex of genes, right? So HLA-A, HLA-B, HLA-C, HLA-F, and each one of them can take on slightly different properties, and they form a multi-protein complex that gives us a lot of variety, right, in this complex. That's seen here, and I don't think I have time to cover all this. Otherwise, we're not going to get to the assignment. But our HLA-B gene is here in the HLA-1 complex, because we're not talking about the class 2 and the class 3 proteins. Super complicated thing. And you may have seen these allele designations or haplotype designations, HLA-B, and then a bunch of numbers after that. And this is telling you what some of those numbers refer to. Specific types of proteins, specific changes, specific sites that have changed. So the particular allele group. And we were interested in the allele 15, right? HLA-B 15:01. All right, so that represented a particular allele with a particular variant within that allele. And then what I've assigned you are 7 different variants. So each one of you has one of those variants that you're supposed to find out more about. Those are individual variants, not haplotypes, not groups of variants. You're looking and going to hone in on one single site in the human genome and identify what kind of change that represents. All right, are we ready? Are we ready to get into it? Yeah, the super geeky molecular genetic stuff, it's coming up. Here we go. All right, got my video on. Let's get myself set in the right spot here. Okay, that's good. I should be out of the way. And we're here at the National Center for Biotechnology Information. And what I suggest is the way to start, and there's many different ways you can get to the same information on this site, right? There's lots of databases, there's lots of links. This is truly the web out there. But what I suggest is you just type in HLA-B. HLA-B, that's our gene of interest. And we're going to search for it. Hopefully this internet connection holds up here. And here we go. And it suggests that we're probably looking for HLA-B, the major histocompatibility complex class 1B gene. And this happens to be from Homo sapiens. Actually, HLA is usually the name of the gene in humans. We're going to do some sequence comparisons, and you're going to find out what this similar gene is called in other organisms, because other organisms do have a major histocompatibility complex gene. All right, so we could go and look at a variety of different and learn lots about this particular gene, right? We could go to PubMed and look at the 15,000 different papers that have been written that either mention this gene or about this particular gene. We could go down here and look at a genome, the human genome, and find it there. That's one way of getting there, but I'm going to show you an easier way. We could look at the 26,000 sequences that include the HLA-B gene in them. We could look at clinical variations, population biology data sets. All right, you get the idea. There's a whole lot of things we could explore. So we're scratching the surface with this particular assignment in just getting you to experience some of these data sites that are freely available for anyone to use and explore. So there's a tremendous number of resources here available to you, and anybody who has access to a computer can access this particular site. People from all around the world are using it right now. But I'm going to show you the easiest way to do it and what I've told you to do in the assignment. You can go to Genome Data Viewer right here. So let's click on that. Genome Data Viewer. Here we go. Alrighty. What are we looking at here? What are we looking at here? We're looking at Homo sapiens, that's us. The data viewer, we're looking at a particular assembly of the human genome. And right down here, we've got this little map over here on the side that shows all the chromosomes, right? You've got your 22 chromosomes plus your X, your Y chromosome, and even your mitochondrial DNA, your mitochondrial genome, all represented here. And if I clicked on any of these, I could then go to that particular chromosome. It would show me a map of that chromosome, and then we could dive into that chromosome and look at the genes that are in that chromosome. But what it's done is it said, like, we already know you're interested in HLA-B, right? Because that's where you were at in your first search. And then I'm taking you to that particular gene in the genome. And so what I have laid out in front of me on this row up here is that is chromosome number 6, because that's where HLA-B is found. All right, so chromosome number 6. And we're on the short end of the chromosome. So here we are, this little, I don't know if you're able to see it, but there's a little blue bar right here. It's very, very skinny. And that's because this is, I don't know what the size this is. As we'll scroll in, maybe we'll find out. But we're talking tens of millions of base pairs long. But our gene's only 3,300 something. All right. And so that little tiny line represents where the HLA-B gene is. And then below here, it's saying, okay, now here's the segment. It's actually from base pairs 31,354K. So that's 31,354,000. You know, running down over here to 31,357,000. Which would be 3,000 more. So I said the gene was a little over 3,000 base pairs, and so that's what you're seeing here represented. And then you see these little green boxes, these dark green boxes. Those represent the exons, the express sequence, the actual code for this gene. I'm sorry, going to get lots of pop-up boxes because nearly everything on this page is access to additional information. Right, so almost anywhere I land with my mouse, it's going to like pop up a little thing. It's going to like say, here's all kinds of other information, and there's lots of highlighted links there, which you can take to find out about the database that shows you that particular sequence. All right, so let me find a place where that's not going to happen. All right, so the green boxes are the exons, and the other spaces are the introns between those sequences. Now, in your assignment, I told you roughly where your variant is. I didn't want to lead you blind because it's going to be hard to search 3,000 base pairs for your particular variant. And so I told you that your variant is in either exon 5 or the intron 5 or exon 5. And so 1, 2, 3, 4, 5. This is the 5th exon right here. Yeah, see if I can show that to you easily. Exon 5 of 8. I can't scroll up to show you that, but it's in that list right there. It says exon 5 of 8. You might have noticed I counted from left to right, sorry, right to left instead of left to right. And that's because this gene, you see the arrows in these green boxes? They're pointing from right to left. Right, that indicates that the gene is read in that particular direction. So on this particular portion of the chromosome, these genes, or at least this gene, HLA-B gene, is actually read from right to left, instead of the typical left to right that you would hope to see usually. But we've talked about how your genes, are read in both directions. And because you can have sections of your chromosome that are turned around, right? Inverted. And when they're inverted, the genes just get turned around. They're facing the opposite direction. So this is one that's facing the other direction. That's why I counted from right to left. I picked out the very first green box. And I said that's exon 1, and then exon 2, exon 3, exon 4, and then I'm over here on exon 5 now, out of the 7 exons that there are. So we want to look at exon number 5 and exon an intron number 5, which come right after exon number 5. So that's the space in between exon 5 and exon 6. So how are we going to look at that? Right? What we're going to do is I'm going to hit this little plus sign here, and we're going to scroll in. All right, so now here's exon 5 right here. And let's say you want to move down. I can use a little arrow and I can move down the human genome here and back and forth, right? So here's exon 5, here's intron 5. And now if I scroll down, I start seeing some things down here because I'm getting a little closer to the sequence. You see, now we're at 31,354,800. This is 31,354,900. So this is only 100 bases right here. So 100, 200, 300, 400. It's about 425 bases between these two exons. So that's how large the intron is, intron number 5. And I've given you that sequence, right? I've shown you in your assignment, I have copied out the entire sequence of this whole thing. So once you find your mutation, right, your variant, you're going to be able to go to that sequence, and I'm going to have you mark it on that sheet. So you'll start seeing, okay, here's RS25238,607, and that is either a T, an A, or a C, right? You see where my arrow is? It says that that particular variant is either a T, an A, or a C. So that's a place where we differ as human beings. One of you out there might have a T, And I might have an A and somebody else has a C. But apparently a G is extraordinarily rare or doesn't exist in the human population. All right, these are all common, or relatively common variants. Over here we got RS 2523608, and that could be a G, an A, C, or a T. In other words, you could have any base there, right? Some people have one, some people have another. But we got to keep scrolling in. Now you might get lucky. Your variant might show up right here. These tend to be some of the more common variants that show up first as we're sort of zooming in, because honestly, there's not enough room to show you all the variants right now. There's a lot of variants along the sequence. Out of 400 base pairs, you could expect that in an intron, there's going to be dozens, if not 100 different sites that might vary if we looked at thousands and thousands of individuals. So let's scroll in a little bit more. So I go up to my little plus and I zoom in. Now I've got exon 5. Let me just confirm that. Yeah, we're still in exon number 5. And I'm gonna, well, I don't want to scroll over too far. All right, now I've lost myself. I got to go back. Right, okay, I forgot I could drag over too. So let's drag over here. Now I'm going to scroll in. And then I'm going to scroll in a little bit more. And so now here we've got exon 5. And now I can see that exon 5 is, you know, we're at 31,355,100 up to 31,355,200. So each one of these segments is, well, 40 to 60 to 80 to 200. These are only 20 base pairs each. So we're getting really close to seeing the raw sequence at this point. And now if I come down here, I'm seeing, well, there are some more variants, but let's just keep on scrolling in until I see the sequence. Ah, there we go. All right, now we're down to 10 base pair intervals. So we've zoomed in all the way down to the sequence. And now I can see we're in exon 5, and I can drag this down here, and I can see exactly where the end of exon 5 is right now. All right, it ends at this base pair right here. So I could count 31,355, 100. So it's 100, 101, 102, 103, 104, 105, 106, 107. 31,355, 107. That's what base pair position it is on exon number 6. All right, so now I'm going to scroll down, and now look what we've got down here. This is a whole list of variants. Each one of these is at a different position. And so let's see. Let's pick out variant RS1766796480, which is either a G or a T. So that one is either a G or a T. And I look and I see G or a T. This is a little hard to do. Yeah, so we've got this G up here. That can either be a G or a T. If it's a T, of course, it's not going to be across from a C, right? The G pairs with a C, but if the G changes to a T, then you're going to have an A across from it, the complement from it. Right, because the pair changes, not just a single base pair. The original mutation was a single base pair mismatched, and we talked about the whole thing with that mismatch, and if it doesn't get repaired, then it gets copied again, and then eventually it turns into a pair that both of them are different than what was there originally. But in this case, you're going to see that in the sequence, either GC or you're going to see TA at that position, because it's the G or the T that changes. All right, so that's our location. So now you found the location. If this was, I'm not doing one of yours, right? You got to find yours. You're going to have to scroll along here and look for your particular variant. And once you identify where it is, you're going to look up here and you're going to identify the exact location on the sequence, 31,355,100, and that's probably 108. Actually, what did I say? The GC? That's 9. 109. And keep this in mind. Here's the sequence. So now I say, I want you to take a screenshot of this, right? That's one of the first things you put on your worksheet, right? I want you to screenshot this page so that I can see that you have the sequence up here and you have your variant down here. It'd be really nice if you like circle that variant too, or put a little arrow there like, yeah, I found it. There's my variant right there. Now you know where it is. And so from there, Now what you're going to want to do is you're going to want to go to your worksheet and go down to the bottom of the worksheet where I've actually listed the sequence of the gene itself in the GenBank form. So let me go out and let's show you that real quick. All right, so here's our assignment. Yeah, here we go. You're picking your variant. Please show a screenshot photo from the browser page showing the location of your variant. That's where you're going to pop that in right there. And then you're going to identify the variant position on the copy of the sequence provided below. You know, on what base pair is that sequence found? So you're going to identify, oh yeah, it's right at this particular base pair, that particular number. And then you're going to design your primers from that. Well, how are you going to do that? Well, you're going to have to go down to this sequence and you're going to have to find that sequence in here again. Now, what I suggest to you, so you're not just spending a lot of time just searching this, is go up here and you're going to look for the directions as to where you can find exon 5. So let's see, exon 5 is right here. Oops, sorry, exon 5 is right here, right? It says exon 5 is 2,252 to 2,368. That's HLA-B, exon 5. All right, now exon 5. Yeah, what happened to my sequence? I know what happened to that sequence. Now that was an interesting OneDrive effect. Right? I have this file on OneDrive and on my other computer at work, because I'm at home right now. The other computer at work, I separated the directions from the worksheet. I put them in separate files, right? And then I renamed that other file. And so now that I came home, I had this file open already on my computer. And so when I scrolled down and I started looking at it, what it did was it updated and it synced the file. Well, I had taken this portion out and cut it out. And so what it did was it synced and it discovered like, oh, this sequence doesn't really exist. So that's why it just disappeared in front of our faces right now, which means I have to go back into OneDrive and I have to pull up that file. So let me do that. Hopefully I'll remember to cut this section out so we're not wasting time with me trying to find the other file. Okay, we're back. All right, so there's the sequence. It tells us where it is. Let's not waste time figuring out the exact site. It told you the exact base pairs. Let's just say theoretically that it was somewhere in here and you said like, all right, you highlight that section. So like this part right here, that was exon 5. And we knew that it was like the third base into exon, actually it was the third base from the end of exon 5. Ah, tricky. And this is why you should be watching this video, right? Because someone who doesn't watch this video and tries to do it themselves, probably going to be confused and get this wrong because they didn't realize that the gene is going backwards when they see it on the genome browser. So they're going to be looking on this sequence, which is in forward direction, all right, reading from top to bottom through the different exons. And so when they look at this, their sequence is actually here at the end, but they think it's at the beginning if they're reading it on the website. Right? I don't know if that made any sense, but somebody's going to be confused. So here, I'm here to show you that you're going to select your sequence, and then you need to go look at that genome browser site, and you need to look at the sequence on either side of your variant, and you need to come in here and find it. And let's say it was this G right here. All right. And so you look and see the sequence. Oh yeah, I see this sequence. It's on in the case on the genome browser, it's on the left side. All right, and then I see this sequence on the genome browser on the right side, on the other side of that particular variant. All right, so you're going to highlight for me, somehow indicate like, yeah, there it is. I found it. I found it in this sequence on the answer sheet, right, that you're going to hand in. And now I'm asking you to develop primers to amplify a segment of this whole gene. But I want it to be a piece that's 700 to 1,000 base pairs long. So here's 2,100 right here, 2,101. And here's 2,700. Actually, let's go to 2,821. That would be 720 base pairs, right? So that's within the realm of what I'm asking you to find primers for, to amplify a segment that large, and it contains the variant of interest. Your variant has to be inside your PCR product because we're developing a genetic test to test whether you have that particular variant or not, or which particular version of the variant you have. So it needs to be between 700 and 1,000. So if you blocked off that 720 base pairs, you could say like, well, all right, I'm going to make a primer here. That's going to be my forward primer, say these 20 base pairs. And then you're looking for something somewhere down here that's 20 base pairs, 18 to 24 base pairs. That's going to be your reverse primer, which you will remember to do in reverse complement, right? Made a big deal about that in class. The previous assignment where you made primers for ABO, a number of people didn't make them in reverse complement. They simply looked at the sequence, they wrote it out, and they said, oh, here's my reverse primer. It's this. Or they thought they got clever and they wrote out the sequence T, C, T, 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 A, C, A, G, G, because that's in the reverse direction. But it needs to be in the reverse complement. If you're going to copy this sequence, you need to have the complement of this sequence, which would be G, A, T, G, T, C, C, because that's 5' to 3', and your DNA polymerase then will copy the complement of that sequence as it goes in the reverse direction. The forward primer, of course, can be exactly the sequence you're seeing right in front of you now. And so it's going to read T, G, A, G, 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 so forth. So that's what you're going to put on your answer sheet. You need to write out your primers. They need to be 5' to 3' the way they're written. They need to correspond to locations that will amplify 700 to 1,000 base pairs of sequence. But not only that, you better make those primers, or you should be making those primers so that they would work efficiently. It's not simply a matter of guessing, right? And I think you got that idea from the last assignment. And I give you the whole set of directions for that, right? Here they are. Rules for designing primers, right? So make sure you look through this list of rules for designing primers in terms of getting the melting point temperatures similar between the two, the length of the primers, if they're compatible with themselves, whether they can fold back on themselves, whether they make primer dimers, whether they attach to each other, right? You need to look at all those things and ask yourselves, would these primers work in real life? And that's what we want to assess. Like, are you able to recognize that these primers would probably, or predict that they would probably be able to amplify that correct for it? We're not actually going to order these primers and use them, but that would be the ultimate test, right? Like ordering your primers, and we don't want to waste a bunch of money if we were going to do that and order primers that don't work. So you're going to design your primers, and you're going to say like, okay, I've got these primers. They can amplify this segment. So let's say we're, from here down to here. That's what they're going to amplify, from the forward primer to the beginning of the reverse complement primer down here. And so that's your segment of DNA you're going to amplify. So we say if you were really to do this, you would amplify this segment, you make millions of copies of it, and that would be your PCR product. And then the next step I'm asking you to do, and in stage 2 of this assignment, is to take this particular sequence, and you're going to take it to a restriction site, all right? A restriction enzyme cutting site. That's not the word I'm looking for, but don't take that sequence. I've given you just the straight up raw sequence without numbers on it. And so you need to find your sequence in here, right? So you look at the beginning and the end of it and go down here. It's in the same order. So you should be able to find it in here. And then once you've found it, you're going to cut it out of this section so you don't have spaces in the numbers. So you can copy this, and then you're going to go find your restriction digest cutter. So let's see here. Restriction site mapper. Yeah, I've looked for that before. And there's a whole bunch of them. And I don't care which one you use. So you might find that one is like the output is difficult to understand, and so go try another one. I'm not forcing you to use any particular one, but hey, I'll just go for number 1 right here. And so it's pretty simple. You're going to select all the possible restriction enzymes. So they give you a list of all these different commercially available enzymes. And you're going to paste your sequence in here. I'm not going to paste the sequence in here. I'm going to just type some random sequence. I'm just doing A, T, C's, and G's. And so I'm making this as random as possible, but with some repetitive patterns in it. Right? Because you will have repetitive sequence, especially in your introns. Oh, I've got to get some more C's in here. All right, that's probably enough to get you the gist of what's going on. But you're going to put in your entire PCR product. 700, 750, 850, 900, however many you came up with. They can be reliably amplified with your wonderful PCR primers. All right, so now that I've got some sequence in here, I want to map this. You can also do a virtual digest, which will kind of show you what it would look like. But let's map this out. And so, well, okay, we got, here's our result. Non-cutters. Look at all those. Those are all non-cutters. What non-cutter means is those enzymes didn't cut your sequence at all. Even though I typed out 100 base pairs randomly, they're all of those restriction enzymes, which remember, a restriction enzyme looks for a specific code of either 4, 5, or 6 base pairs of sequence. But I did not type randomly any of the codes that all those enzymes account for. So they would not cut anywhere. And so if you applied them to your PCR product, that 100 base pair PCR product, nothing would cut, and you would still have a 100 base pair PCR product. However, if you were to apply AVA1, which is an enzyme I've used in the past in one of my projects, pretty common restriction enzyme, it has a restriction digest recognition sequence of CY. I remember Y represents two, I can't remember which two base pairs. It doesn't represent all four. Remember, N represents all four. Like down here, N's represent, it doesn't matter what base pair is there as long as there is a base pair there. Y represents two of them. C, G, R represents a different pair of possible ones, and then G. So apparently the sequence that I typed out had C and then one of the other two base pairs that's allowed to be there, then it had a C, then it had a G, then one of the other two base pairs that had there, and then a G. I'm kind of embarrassed that I can't remember what the Y and then R specifically stand for. I knew that at one time. And so it's a 6 base pair cutter, and it cuts in one location, and that's at base pair position number 61. All right, number 61, that's it. And so if it cuts at 61 and my entire sequence was, how long was it? That must have been 150 base pairs at least. Did I really type in 150 base pairs? If it's 151 base pairs, then site 61 would mean it would cut it into a 61 base pair fragment and a 90 base pair fragment. And if I ran that on gel, that's what I should see. Actually, let's go back. Virtual digest, I haven't done, got an error anyway. I hadn't tried that before. I mean, I haven't tried all the restriction digest mappers, but there's lots of ones that have lots of different interfaces, right? Graphical interfaces for showing what your results would look like. So you can play around with those. Because I'm going to ask you to actually kind of draw out, what do you think your gel would look like if you cut it with this particular enzyme? What would it look like if you cut it with this enzyme and you had the A version at that variant site? And what would it look like if you had the C version at that site, whatever your particular variation is, whether it's a deletion, an addition, or a single base pair change. And that, of course, would either cut or not cut. And so that would give you the differences. And that's what would make it sort of a genetic test, right? You could identify whether you had that particular base pair there or not. All right, so that's the restriction site mapper. And what you're trying to do is you're trying to find, let me do this again. You're trying to find a restriction enzyme that cuts at your specific base pair. So you're gonna have to look at your whole PCR product. You should know exactly what position you're looking for. I'm looking at position number 352. At position base pair 352, that's where my variant is. And you want to confirm that when you look at the restriction site sequence, recognition sequence, that, it was this C right here. That C is the variant site. You either have a C or you have a different base pair at that position. And so if you had the C, it cuts. If it is the T, the other version, it won't cut. And so that's going to allow you to see the difference. Right? So you need to find the cutting site. And as I said in class, or I will be saying in class to you, it took me a long time to decide what variants to assign. That's because I had to test every variant to make sure that the variant was an actual cut site. So all the variants are ones that the sequence will cut when you take it out of the Human Genome Browser. And so it's the other variant, the other version that doesn't get cut. All right. So you should be able to find, using that sequence from the human genome, right, the GenBank file that I'm giving you, you should find the cut site. And so you need to identify what enzyme cuts at that particular location. And then you need to, well, just go through and answer the questions that I have for you. And it basically is asking you a series of questions about like predict the fragments that would result if you cut it. Now, you're not always going to cut it at that one site. You notice that this one down here, AVA 2, it cuts at two different sites, at base pair 58 and at base pair 93. So in that case, you're going to end up with 3 fragments, right? You had 1 fragment that you cut in 2 places. You're going to have with 3 fragments when you're done. You might discover that your restriction enzyme, because remember, you're looking at 700 to 1,000 base pairs. This was just 150 base pairs. So if you have 1,000 base pairs, you might find that you have a restriction enzyme that cuts your location of interest, but it also cuts at 7 other locations. And presumably, we're gonna, for the sake of this particular project, we're gonna assume that the other 6 sites, right, where it cuts, besides the one you're interested in, that they're invariant. They would cut you, they would cut me, they would cut anybody in the world. So they're not of interest. They're not a variant that would be different between human beings. We're just interested in this one site. However, you need to know the pattern, right? So if it cuts 6 times, you'll have 7 fragments. And when you run those on the gel, right, the largest fragments at the top, the smallest ones at the bottom, then you can kind of like draw out like what you would expect it to look like if you ran that gel. All right, so what else are you going to do? Well, I'm also asking you to do a blast search. I want you to explore something about this sequence. I'm going to pick a different sequence than you're going to use. So let me go up here and let's just pick out something in here. This is going to be near the beginning of the gene. And I'm going to pick out, each row is 60 base pairs. So 1, 2, 3, 4, 5, 6, 7, 8. 8 times 6, that's 480 base pairs. Let's check out this 480 base pairs. All right, I want you to copy that sequence. Then we're going to go back to, we're not going to Genome Browser. Let's go back to NCBI, the home page. And now we're going to go to BLAST. All right, we come to this page, and I've given you the link to this page as well. So this takes you to the BLAST tool, and there's a variety of different ways you can BLAST, which is take an unknown sequence, or in our case, it's a known sequence, but we're going to compare it to other sequences. We're going to ask, what other sequences in the database are the most similar to the one we're putting in? And we're going to do nucleotide BLAST because we're comparing sequence to other sequence. Right? We could take translated nucleotides and compare those to other proteins. And we could take protein sequence and compare it to protein sequence, right? There's a bunch of different databases here and different ways to compare nucleotides to proteins, proteins to DNA, and so forth. But we're just doing DNA to DNA, so nucleotide BLAST. I click on that, and so we're in BLAST N, nucleotides, and I'm going to paste my sequence in there. All right, so there we go. There it is. Doesn't matter that there's returns, all right? There's my sequence, my 400 and some base pairs that are in there. And then you're going to use the defaults for most everything here, except that I'm going to ask you to go down here and type Homo sapiens, which is taxa 9606. And don't forget to do this. Many people did on the last assignment, which you did something similar. I want you to exclude this, right? We want to exclude human beings from our comparison because we know we have a human gene. You might think, well, it might be interesting to look to see how different this sequence is from other humans. But I can tell you, it's not going to be too exciting. You're going to get 100 sequences for just those 400 base pairs, and they're going to be 99% similar, if not 100% similar. Actually, you're probably getting 100 samples that are 100% similar. There's probably 100 people in the database because there's a lot of sequences out there. There's probably 100 people that have identical sequence. So it's going to come back with all those first. And so that's going to be a boring list. Yes, it's true. There are variants, right? There are people who have different sequences in those 400 base pairs, but you're not going to find them because it's going to come back with the most similar sequences first. We might have to, if you wanted to do this, you'd probably have to come down here and go like your max target sequences. I want 5,000 results. That way I can scroll down 5,000 and see like when do I finally find some differences. All right, that's not going to be that great. All right, so you've got you've excluded Homo sapiens, and I'm gonna say choose more dissimilar sequences. Did I say that in the assignment? Paste 250 base pairs of sequence. highlight the variant location. Sorry, I'm just going to make sure I'm doing the right thing for you here. No reason doing a demo if I'm demoing the wrong thing for you, am I? Yeah, 9606, under program selection, click the radio button for somewhat similar sequences. Ah, yes, somewhat similar sequences. All right, blast in. Somewhat similar sequences. And click the algorithm parameters and change max target sequences to 250. All right, so this is the algorithm parameters, and we're changing to 250. All right, lots of other things we could change, but there's no reason to. We're just going to go blast this thing. All right, and away we go. This page will be automatically updated in 12 seconds. Dun, 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 dun. Now, I'm doing this at 10 p.m. So that's 7 p.m. Eastern time. It's pretty much dark in Europe. I mean, I should really have good access to this this supercomputer that we're using right now. So I'm a little surprised by the whole 17-second delay here. I was expecting 2 seconds and 5 seconds and 7 seconds, 10 seconds, and then boom, we'd have our results. But now it's saying 22 seconds. Depending on what time of day you're doing this, you might find the server is more busy than others. Also, we asked for somewhat similar sequences. It's actually going to, it's not looking for exact same length of sequence and matches. It's willing to sort of like break up the sequence into a little bit, separate parts and say like, well, maybe what you're comparing to has an insertion or a large deletion. So part of your sequence matches to this and part of it matches to something else, like farther downstream. And so that requires a lot more pairwise comparisons, right? It's a lot more computation to do somewhat dissimilar than it is to do like find the most similar sequence or only really similar sequences, because then it just starts excluding giant chunks of the database to look at because it immediately discovers that it's not similar to a lot of things. And then it quickly finds a human sequence that's almost identical. It's like, yeah, let's look at things that are similar to that. And it starts working on that. All right, lots of different parts of I mean, to me, it's all a black box. I don't really truly understand the math of this whole thing. Wow, we're looking at 30 seconds now. How am I going to fill this time? Let's use it wisely. All right, so the last step, what we're doing here is we're waiting for our comparison, and then it should come up with a list of different taxa, different organisms, and it'll tell us what gene is similar to our sequence that we just put in here. And it's going to tell us how similar it is. And so I'm going to ask you how similar it is. And then the step that I'm going to take you further than you have in another assignment that you've done with bioinformatics is I'm going to ask you to do the multiple sequence alignment. And that is going to take the results and it's going to align the sequences for you. And it's going to help you visualize where the differences are between those sequences. And so I'm hoping to see some interesting patterns. I'm hoping to show that to you right now. Right now in 30 seconds. All right, we're almost to our 30 seconds. I'm hoping for results this time. I don't know what to do if this thing gets stuck in some kind of permanent loop. How about while it's working, I open up another one. Let's go to blast, nucleotide blast, paste that in there, exclude homosapien. That actually remembers my parameters. 250, hit blast. Let's get a second one going at the same time. That'll be done in 2 seconds. This one still says 30 seconds. See which one comes in first. Come on, National Center for Biotechnology. My tax dollars are working hard here. Actually, I guess they're not working very hard right now. But I did not anticipate this being a problem. I did test this before. I know it doesn't seem like it, but I did test this. I've gotten results, and that's why I wrote it up the way I wrote it up. All right, you want to see what happens if we look for humans? Let's go to blast, put my sequence in. It could be the section of sequence I picked was more variable. Not going to exclude humans. And let's go for highly similar sequences. Let's try that. I might just have to walk away and then come back and when it starts working. All right, I'm going to pause again and go check my email. All right, we're back. I mean, it took 14 minutes, but we finally have results. I have results for the first search I did. Second search is still hunting after 8 minutes, 6 minutes here. But the one in which I said I wanted to compare to humans with similar sequences, I came back in 3 minutes. Let's take a quick look at that one real quick. So if I scroll down here, it's like, oh, shocker. My sequence that I put in is most similar. 100% query coverage. That means all the bases that I put in, it covers that whole, and it's 100% identical. And guess what? It's Homo sapiens, MH class 1 antigen, HLA-B gene, HLA-B 49, 01, 01, 01 allele. That's the actual allele that I'm giving you in the assignment. So it's the sequence. It came back and found itself, which is a good sign. I mean, it should find itself and it should be identical. And then it's 100% similar, 100% similar, 100% similar to 4501, 5002, 5004, 5407, 44.15. All right, these are different alleles of the HLA-B gene. Keep on going, keep on going, keep on going, keep on going. These are 100% similar, 100% similar, 100% coverage. The different cell lines. Oh, now they're 99.81 similar. So there is a HLA-B 40 colon 30. That particular allele is 99.81% similar, which means it covers the same base pairs, but it must have one difference. One must have one base pair difference somewhere in there. Keep on going down here. All right, so these are all the same gene. Every single one is the same gene. And even after 250 results, they're still 99.81% similar, only one difference between them. Completely, it's 540 base pairs long. All right, let's go over the more interesting one. So let's scroll down here. Remember, we excluded human beings. All right, so since we excluded human beings, our first result it's most similar in this case to gorilla. And it's similar to the gorilla GOGO-B gene, which is just the name of HLA-B gene in gorillas. And then here's pan troglodytes, which is chimpanzee. And that's PATR, all right, pan trog. And that is, and it's the B version of their gene. So this is the major histocompatibility complex 1 antigen B gene. So it's the same gene. They just give it a name, a chimpanzee name. And then we've got pan troglodytes, pan troglodytes, pan troglodytes, gorilla, gorilla, pan troglodytes. And the reason they're interspersed with each other is because out of the 540 base pairs, they have, looks like generally 100% query coverage, meaning they found 540 base pairs that aligns with 540 base pairs of human being. And the percent identity is 96.86, 96.86. So we could click on one of these, and that will take us down to where the alignments are. And so our sequence compared to the most similar sequence in the database, although I would say the top 50 here are essentially the same similarity. It's just like this one has this difference and this one has this one other difference. And so I compare these. Yeah, I say, well, actually, it's more than one difference. 524? Oh, sure, it's going to be more than one difference, right? Human beings was 98 point something. This is 524 out of 541. So there are 17 differences between the human sequence and gorilla sequence. And I can see some of them in here. There's a T, there's a C in human being, and then there's a T in the gorilla right there. And there's a C and a G here, where there's a T and a C there. There's a G over here and an A here. You get the idea. All right, but it's mostly individual nucleotide changes. Oh, here's one. There's a base that human beings don't have, but gorillas have a G there, so they have an extra base pair. And you might be thinking to yourself, oh, frameshift mutation. They've got an extra base in the protein coding sequence. But we didn't actually look at what sequence we pulled out. I want you to know what sequence you pulled out, because you're going to know where the intron and the exon are. I may have pulled out mostly exon sequence. I'm sorry, intron sequence, which is between the genes, in which case having insertions and deletions really doesn't affect anything. And so those are probably not a meaningful difference. In fact, most of these differences aren't actually meaningful differences, I would suspect, between us. All right, now I can scroll back up to the top here. We could look at graphical summary. That's going to be kind of boring. Like, yes, there's lots and lots of sequences that have really, really high similarity compared to human beings. What we want to do is we want to look at not the alignments. We want to look at the multiple sequence alignment viewer. That's MSA viewer, which is going to open us into a new page. And this may take a moment because it is taking all the sequences, lining them up. It's going to draw them out for us and do a nice little comparison. Here we go. All right, so what we have on top is that's our sequence from base pair number 1 up to base pair number 540. And these red bars are representing locations where there is variation among the 250 samples we're comparing it to. Now, we could have compared it to 1,000 sequences, but 250 is a good number. And then what we have here is we've got all these ones that say plus troglodytes. We're going to ignore the minuses for now. I don't want to get into the differences there. We're just looking at these ones that have this plus sign on it, and I made a note in that in the assignment. That has to do with the ones I want you to look at patterns. And so as you look across here, these little red hash marks are where there are differences between the human sequence and, in this case, the two top ones are gorillas. And you can see the two gorillas actually are fairly different from one another, right? You see that? I mean, the gorillas aren't identical to one another. If you looked at two human beings, now we could do that by going back to that other sequence alignment I just did, in which we got all human beings, except there was only like 1 or 2 differences total among all 250. So that it would be a kind of a boring graph. This is why I wanted you to exclude human beings. But the point is that you and I are really similar in that short sequence. Whereas gorillas, there's a good amount of variation between these two gorillas. And then we come down here to chimpanzees. There's a lot more similarity in the chimpanzees, at least the ones that they're in the database. Now, I don't know anything about these individual chimpanzees. They might all be from one location. They might be all somewhat related to one another, and that would explain a lot of their similarities. Whereas gorillas, there are sort of lowland gorillas, upland gorillas, and we know that they're fairly genetically different. So likely these are two gorillas from two very distinct family groups that have not had a recent common ancestor. And so they've accumulated more changes between them. So you can see that the chimpanzees are all similar to one another, I would say. And then the gorillas, it's even hard to say they're similar to one another, but they're not the same as a chimpanzee, clearly. And they obviously have differences from you and I. And I could keep going down here and I can see like, oh, look at all these more gorillas here. Oh, now here's some gorillas that are very similar to each other, right? They have the same variants, and a few differences where they're missing this variant. And then we get to, here's some more chimpanzees. And now I'm seeing some variation in the chimpanzees. And then we come down here. Here's macaques. All right. And you notice the macaques are, there are some variants that are similar among all the different. Here's a variant right here. That's the gorillas, chimpanzees, and macaques. They all have a variant compared to human beings. And so the farther you get away from humans and more distant from humans in terms of genetic similarity, you'll start seeing a lot more variants showing up. All right. Boy, there's a lot of macaques in the database. Somebody sequenced a lot of macaques. And then we're back to, here's some sequences that aren't, like maybe they don't have a complete sequence of this particular individual chimpanzee. They only sequenced a portion of it. And because they only sequenced a portion of it, it doesn't get a really high score because you don't have the whole thing to compare to. And that's why it's down here at 200 instead of up there, even though it doesn't have that many differences from human beings where we have the known sequence from that. Okay, so I want you to take a screenshot of this. And then I have a few questions about the patterns that you're seeing here. And you're not going to see exactly this. I picked a different portion of the gene for a reason. I want you to pick something around your area where the variants are. You'll probably see a somewhat different pattern than what you're seeing here. Alrighty. Yeah, that's this last part. From your blast page results, click on the MSA viewer, paste a screenshot photo of your MSA viewer screen below. Zoom in to find your variant. So if you have 1,000 base pairs, you're going to see the overall pattern. That's going to be nice, but you're going to want to maybe zoom in a little bit more so you can see and find your particular variant, because what would be interesting also is I'm going to ask you, does your variant exist in other primates, right? Do they also vary at that particular location? Remember, you're only comparing one human being with all these other primates. It's kind of hard for me to have you look at 50 different human beings and compare to the primates, because it's hard to do the multiple sequence alignment with that, because in this, we're limited in our options here. Let's just put it that way. So I'm having you exclude humans, because I think you saw that if you just did humans or just searched everything, you would get all humans. And that makes for a boring sequence alignment. All right, I think that's it. The rest, I guess the only thing else you need to know is you got to have this done by December 4th. Drop it into Brightspace. We'll take a look at it. If you have any questions, email me. Email Hannah, Mahdi, and we'll help you out. We'll get you through this thing. All right, that's it. I got nothing else to say, so we got to end it here. Talk to you later. Talk to you in class. Bye-bye.